In continuation to the previous lecture of bioorganic chemistry, today we will start mechanism of enzyme action. In most chemical reactions, an energy barrier exists that must be overcome for the reaction to occur. This barrier prevents complex molecules such as proteins and nucleic acid from spontaneously degrading and so is necessary for the preservation of life. When metabolic changes are required in a cell, however, certain of these complex molecules must be broken down and this energy barrier must be surmounted. Heat could provide the additional needed energy called activation energy, but the rise in temperature would kill the cell. The alternative is to lower the activation energy level through the use of a catalyst. This is the role that enzymes play. They react with the substrate to form an intermediate complex, a transition state that requires less energy for the reaction to proceed. The unstable intermediate compound quickly breaks down to form reaction products and the unchanged enzyme is free to react with other substrate molecules. Only a certain region of the enzyme called the active site binds to the substrate. This figure shows the active site is a groove or pocket formed by the folding pattern of the protein. This three-dimensional structure together with the chemical and electrical properties of the amino acids and cofactors within the active sites permits only a particular substrate to bind to the site, thus determining the enzyme's specificity. The active site of an enzyme is a groove or pocket that binds a specific substrate. Enzyme synthesis and activity also are influenced by genetic control and distribution in a cell. Some enzymes are not produced by certain cells and others are formed only when required. Enzymes are not always found uniformly within a cell. Often they are compartmentalized in the nucleus, on the cell membrane, or in a subcellular structure. The rate of enzyme synthesis and activity are further influenced by hormones, neurosecretions, and other chemicals that affect the cell's internal environment. Now, the factors affecting enzyme activity. Enzymes are not consumed in the reaction, they are catalyzed and can be used over and over again. Only a very small quantity of an enzyme is needed to catalyze a reaction. A typical enzyme molecule can convert 1000 substrate molecule per second. The rate of an enzymatic reaction increases with increased substrate concentration, reaching maximum velocity when all active sites of the enzyme molecules are engaged. The enzyme is then said to be saturated, the rate of the reaction being determined by the speed at which the active sites can convert substrate to product. Enzyme activity can be inhibited in various ways. Competitive inhibition occurs when molecules very similar to the substrate molecules bind to the active site and prevent binding of the actual substrate. Penicillin, for example, is a competitive inhibitor that blocks the active site of an enzyme that many bacteria use to construct their cell walls. Non-competitive inhibition occurs when an inhibitor binds to the enzyme at a location other than the active site. In some case of non-competitive inhibition, the inhibitor is thought to bind to the enzyme in such a way as to physically block the normal active sites. In other instances, the binding of the inhibitor is believed to change the shape of the enzyme molecule, thereby deforming its active site and preventing it from reacting with its substrate. This latter type of non-competitive inhibition is called allosteric inhibition. The place where the inhibitor binds to the enzyme is called the allosteric site. Frequently, an end product of a metabolic pathway serves as an allosteric inhibitor on an earlier enzyme of the pathway. This inhibition of an enzyme by a product of its pathway is a form of negative feedback. Allosteric control can involve stimulation of enzyme action as well as inhibition. An activator molecule can be bound to an allosteric site and induce a reaction at the active site by changing its shape to fit a substrate that could not induce the change by itself. Common activators include hormones and the product of the earlier enzymatic reactions. Allosteric stimulation and inhibition allow the production of energy and materials by the cell when they are needed and inhibit production when their supply is adequate. This is the flow diagram for the preparation of enzymes by plant cell, animal organs, microbial fermentation and plant latex. Now the method of enzyme purification. First is salt precipitation and second is chromatographic method. In chromatographic method, first is ion exchange, which is cationic or anionic. Cationic or anionic separation based on charges. The greater the net charge, the more strong the interaction. Evaluate by increasing ionic strength or of buffer or altering pH. Second is gel permeation, size exclusion chromatography. This is a separation based on the size larger protein evaluate first. Third is hydrophobic interaction chromatography. Separation based on hydrophobicity of protein surface elute by decreasing polarity of buffer. More hydrophobic bind more strongly and elute first. And fourth is affinity chromatography. In this chromatography, separation is based on specific biological interaction. For example, enzyme substrate recognition, evaluate with substrate. And fifth is immunoaffinity chromatography. In this chromatography, separation is based on the specificity of antibody recognition of peptide sequence. Elute by lowering pH to 2 to 3 to disrupt antibodies peptide interaction. Neutralize quickly to avoid loss of activity.